Lesson 2, Chapter Biotechnology Principles and Processes. So students, in the last video you have learned about what is biotechnology and what are the various principles that is used in the process of biotechnology. And there we came across the first principle, that is genetic engineering, where the manipulation of the genetic material is done in order to obtain a desired product or any kind of processes or service that can be useful to human beings. In today's video, I will discuss about the various tools that is used in genetic engineering, or you can say that the various requirements that is required for bringing out the experiment where the manipulation of the genetic material is done. So let's start with tools that are used in genetic engineering. And the first tool that I'm going to discuss here is about the various types of enzymes. So this is an enzyme dependent process. So here I'll group some types of enzyme that are used right from the beginning for genetic engineering. And the first type here, this is the lysing enzyme. Lysing enzyme, as you know, the word lyse, it means breaking down. So here these enzymes, they are used to open up the cell to get DNA for the genetic experiments or for the genetic material. And these enzymes, they will help in extracting the genetic material from the nucleus. And one of such enzyme is lysozyme. So lysozyme, it is used to dissolve the bacterial cell wall. Then for different types of eukaryotic cells, the type of enzyme that can be different. Say for example, in case of a plant cell, there the lysing enzyme, it will be cellulase, and for the fungal cell it will be chitinase. And for animal cells, there are various kinds of enzymes that are used from the group lyses. So this is about the first category of enzyme that is used in genetic engineering. Now let's come to the most important enzyme, that is restriction enzyme. The restriction enzymes are used to break specific DNA molecules. And the discovery of the restriction enzyme in a bacterial cell, that helped genetic engineering in many processes. Now, in a bacterial cell, generally these bacteria, they use the restriction enzymes for cutting the extra DNA part from the bacterial DNA. And these restriction enzymes, they are found only in case of prokaryotic cells like bacteria. Now these restriction enzymes, they are of two types. One is called as exonuclease and the other one that is called as endonuclease. Now the difference between these two types of nuclease is that exonuclease, here the nuclease cleave or cut the base pairs of DNA at the terminal ends. That means they will always cut the DNA segment from the end, and it will have only a single breakage point. And the second type, that is endonuclease. So endonuclease, they can cleave or cut the DNA at any point except the terminal ends. That means they always cuts the DNA from the inner part, not from the terminal points, and for that they have two breakages at both the DNA parts or both the DNA segments. So there are many other differences in between exonuclease and endonuclease, such as exonuclease, they act on single strand of DNA and endonuclease, they cleave one strand or both strands, so that I have already said. And you can add one more, that exonuclease, they do not cut RNA, and endonuclease, they may cut the RNA segments. Now here in genetic engineering, a special type of endonucleases is used, that is restriction endonuclease. So it is called as restriction endonuclease because these enzymes cuts DNA duplex, that is the double-stranded DNA, at a specific point. So they always have a specific point, and these specific points, they are called as the recognition sequences. So these recognition sequences, they are the sites where the restriction endonuclease cuts DNA. So the restriction endonucleases, they are also called as molecular seizures. Now when the restriction endonuclease, they cuts the DNA, we get two type of ends. One is the sticky ends and other one is the blunt end. So you can have the example of sticky end and the blunt end with the example of a restriction endonuclease enzyme, that is EcoR1, that cuts its specific sequence in the DNA, that is GAATTC. So they recognize the base sequences at the palindromic sites in a DNA duplex and cut its strand. As you know that the restriction endonuclease, they cuts the base sequence at the specific site. Now here a word is added here, that this sites, that is called as the palindromic sites. Now as I have given you the example of EcoR1, so here you can go to the recognition sequence, that is GAATTC, and as you know that GAATTC, so that is only one part of the DNA, that is from 5 prime to 3 prime. Now definitely there will be the complementary DNA for that, and that will run with polarity 3 prime to 5 prime, as you can see in the picture. So here you can read the other DNA from the 5 prime to 3 prime side, so that will also, there also you will read the same sequences such as GAATTC. So from one side to other side, so from 5 prime to 3 prime, so that also comes as GAATTC. Again from the other strand, that is the complementary strand, so there also you will read from 5 prime to 3 prime, that is GAATTC. So this is just like the word Malayalam. So Malayalam, if you read from first to last and from last to first, so there you will find the same arrangements of the letters. So such type of words or such type of sequences, that is called as the palindromic sequences. So here, as I was saying about the cutting pattern of the enzyme EcoR1. So here you can see that EcoR1, they're in the vector DNA. So there it had cut in the position that is near G in one of the strand, and in the foreign DNA, there also it had cut in the same site, that is G from the 5 prime end, and there we find a zigzag type of or you can say the L type of cutting in both the DNA material. So vector DNA, that is one DNA, and the foreign DNA, that is another DNA. So here EcoR1 was applied, and EcoR1, it will recognize its specific sequence, that is GAATTC. So in the vector DNA also it was applied, and in the foreign DNA also it was applied, and there what you will find that the EcoR1 restriction endonuclease, it will cut both the DNAs there at the G site, and you can see that there is a L patterned gap now, or L pattern exposed from the DNA strand. That means it had cut the DNA in that particular part of the DNA. And we can see the L type. Now when this both DNAs, I can see in the, as you can see in the last diagram, that both this DNA, they can be joined at this particular specific sites. So such kind of ends, that is called as the sticky end cutting, and it gives a L-type pattern by the restriction endonuclease, that is EcoR1. So in that way, the EcoR1, it cuts the DNA in a specific site and it produces sticky ends. And some type of restriction endonucleases, they cuts the DNA right at the middle, and in such cases we get a flat end, and in that we call it to be blunt end. So the restriction endonuclease enzyme such as EcoR1, it produces two types of ends after its cleaving process. So this was about that how the EcoR1, it cuts the or clips the DNA material. Now let's come to some types of some types of restriction endonuclease. So restriction endonuclease, as I have already mentioned one name, that is EcoR1. So that is a restriction endonuclease enzyme that recognizes the site GAATTC from a DNA duplex. Now here, what does this EcoR1 mean? So this sometimes comes in the exam. What, or expand the term EcoR1. So it has a meaning. So Eco, that means it stands for the bacteria Escherichia coli. So from Escherichia comes the letter E, and from the species name, that is coli, comes the second two letters, that is CO, eco. And Escherichia coli, that is the bacteria from where this particular sequence was extracted. Then R, it stands for the strain. So the strain of Escherichia coli that was used, that is RY13. So where the R denotes this RY13 strain of the bacteria or type of this bacteria, and the Roman 1, it denotes that it was the first enzyme to be isolated from this particular strain, that is Escherichia coli RY13. So in that way, there are several groups of restriction endonuclease, and out of that, HIND2, so that is another restriction enzyme which was first discovered restriction endonuclease, that is before Escherichia coli. So HIND-D2, so that was the first restriction endonuclease that was extracted from the bacteria Haemophilus influenzae, and the strain is RD. So for HIND2, here the recognition site will be GTCGSE from 5' to 3', and this is also a palindromic sequence. So similarly like that, there are some other restriction enzymes such as HIND3 and EcoR2, BAMH1, etc. So this was all about the restriction enzymes. So let me repeat just simply that what is this restriction enzyme. They are regarded as the molecular scissors because they can cut the DNA at specific sites, and those type of specific sites are called as recognition sequences, and here the recognition sequence can also be palindromic sequences like GAATTC or GTCGSE. There are similar other types also. And now let's come to the third type of enzyme that is used. So that is DNA ligase. So DNA ligase, that is an enzyme that helps in joining at both the cut base sequences in the vector. So in the previous diagram, as you have seen that two DNAs, that is the vector DNA and the foreign DNA. In both the DNAs, the cutting it was done by the restriction endonuclease enzyme. And these two DNA molecules that needs to be joined, and there the joining it will be done by the DNA ligase. So therefore we call the DNA ligase as the molecular glue. And there are several other types of enzymes also that is used, such as DNA polymerase, then reverse transcriptase. So such kind of enzymes are also used in genetic engineering mechanism. But these are the three important enzymes that is used in the whole process of genetic engineering. Then let's come to the vectors now. Vectors or cloning vectors. Now this is another tool that are used in the genetic engineering process. So these vectors, they are DNA molecules that can carry a foreign DNA segment and replicate inside the host cell. So from the definition of cloning vectors, so what we have come to know that vectors, they are nothing, they are DNA molecule itself which are extracted from some organisms such as bacteria, viruses, or it can be yeast. So such type of organisms are involved. And these vectors, they are used as a vehicle where the foreign DNA segment that is joined and that is allowed to replicate inside the host cell. So you can say that to be a vehicle DNA also. So as you have seen in the diagram that how the vector DNA, so that is the vector DNA where the cloning of the foreign DNA will take place. So for that, the two DNAs from two different organisms, those were joined by the enzyme that is DNA ligase. But there requires some features in the cloning vector. So those features or characters must be present in the cloning vector. And one such feature, that is origin of replication site. So as you know that origin of replication, that is the site in the DNA where the replication starts. And the DNA, it always requires to undergo the central dogma process. So that is why it always starts with replication, transcription and translation, and for that replication it was the first step. So the foreign DNA must be stick or joined into the site where it has the origin of replication. So origin of replication is a specific sequence of DNA bases which is responsible for initiating replication. So there, origin of replication, it must be there. Then comes the selectable marker. The selectable marker, there are some genes that helps in selecting those host cells which contains the vectors and eliminate the undesired gene. So selectable markers, there are also some genes that is present in the DNA of the host, DNA of the vector, and there must be some genes that can help in selecting those host cells which contains the vector or where replication was allowed to be done. Say for example, some such selectable markers are antibiotic resistant genes such as tetracycline resistant genes, ampicillin resistant genes, etc. So here, such type of antibiotic resistant gene, as you know that the bacteria, they possess such type of antibiotic resistant gene. So this antibiotic resistant gene, such as tetracycline resistant gene, ampicillin resistant gene, they are used as selectable marker in order to study the host cell, whether there the foreign DNA is replicating properly or not, and if it is not, then such undesired genes, they are cut out from the vector DNA itself. Then comes the cloning sites. These cloning sites or recognition sites, they are the part of the selectable marker where foreign DNA can be inserted and is responsive to commonly used restriction enzyme. So for GAATTC, there in the cloning vector also there must be the site either in the part of the selectable marker where it possesses the same recognition site. So these cloning sites, they are used or they are the part of the selectable marker where the foreign DNA can be inserted and where it could undergo the process of replication. So for example, tetracycline gene possesses restriction sites for BAMH1. So this is a restriction endonuclease enzyme. Then SUL1, this is also another restriction endonuclease enzyme. And this is tetracycline resistant gene. So it has the recognition sites for both BAMH1 and SUL1. So here this is for the vector that is called as plasmid, as I have named that many bacteria and viruses such as bacteriophages, they are used as vectors. So and the plasmids, this circular DNA, they are present in the bacterial body. So the plasmids, they are used, and these plasmids, they have this particular region, as you can have a look at the diagram here. So this is a plasmid which is called as PBR322. So there the tetracycline gene, it possesses the restriction sites for this particular two restriction endonuclease. And then comes the last one, that is vectors for cloning genes in plants and animals. So generally in case of plants and animal cells, for plants and animal cells, so there the cloning vectors that will be different. So for plant cells, it is always the Ti plasmid that is used, and this Ti plasmid that is extracted or that is found in a bacteria which is called as Agrobacterium tumefaciens. So this Ti plasmid, it gets modified into a cloning vector in case of plant cell if the host cell is a plant cell. And for animal cell, these are retroviruses. Generally retroviruses, they are used to carry the desirable gene in case of animal cells. And lastly, let me name some types of vectors that is used, as I have already named a few now. So plasmid vectors. So what is plasmid vectors? So these are extrachromosomal, self-replicating, usually circular, double-stranded DNA molecule found naturally in many bacteria and yeast. So they are used as vectors which have short nucleotide sequences, and they have origin of replication along with the selectable marker gene. And then comes bacteriophages. So there are many types of bacteriophages or many types of bacteria that is used according to different host cell, and one such vector, that is bacteriophage. So bacteriophage, as you know, that they are viruses that attack the bacterial cell by injecting their DNA into this cell. So the DNA of these two types, that is found in case of bacteria and the other that is found in case of virus, so those are used as cloning vectors, so that the foreign gene that we have bought from a desired organism, so there in these vectors the DNA, it could be replicated. And what is the purpose of replication? That is to multiply the gene or to complete the central dogma process of the foreign gene, so that we can study the nature of the gene. So this is all about today's video, where I have discussed about the first two tools, that is about the enzymes and the cloning vectors. Thank you.